So hello students, welcome back here on channel Vedantu NEET English. This is Basavaraj Sir, your biology master teacher students. Hello, welcome students. Students, if I am audible and perfectly visible to all of you students, quickly fire emoji in the chat to let me know, yes sir, you're audible and visible in the chat, all of you. Quickly some fire emoji in the chat to let me, yes sir, I am properly audible and visible to every single one of you students. Quickly in the chat, all of you. Good evening. Hello Hema, hello Varshita. Good to see students. Last class, I saw all of your beautiful comments, crazy comments. Okay. So hello students, if I'm audible and visible to all of you students, quickly some fire emoji in the chat to let me know, yes, students, if we are ready for today's amazing class. Okay. Okay, students. Now, students, has this ever happened to you? Students, has this ever happened to you that in the textbook they give you a diagram, but this is what they give an exam, and you are not able to tell what is what? Because if this is the case, I will take you to next level today. I will not be showing you a darker version of this. I will be teaching you the entire chapter eukaryotic part in the most legendary way, students. Legendary way. Let me give you a small sneak peek. Okay, let me just give you a small sneak peek on how we will be learning today's entire class. So students, behind me right now is a eukaryotic cell. Now students, I want all of you tell me which one is your favorite organelle. See, mine is obviously chloroplast. What is your favorite organelle? See students, this is your entire eukaryotic cell here. Now in this eukaryotic cell, can all of you see students the entire genetic material? Here we have your nucleolus and the chromatin here inside the nucleus. Then here we have the entire endoplasmic reticulum, which is arising from the outer layer of the nuclear wall here. Then we can see here centrioles. We can see some kind of Golgi body here. Then students, here the almighty mitochondria here, students. So this right here is the entire eukaryotic cell on the board, students. So if you have any doubts, you can always ask the doubts in the comment section. If I look at the doubt, I will clear it then and there. Students, all of you ready today? Can I get a confirmation in the chats that all of you are ready for today's amazing class? Students, let me tell you one more thing, right? If you are anywhere in Chennai, if you are anywhere in Andhra Pradesh, Telangana, Karnataka, we have offline centers offers going on right now. So if you are any part of this entire South India part, please go check it out right now, especially JP Nagar, Bangalore one. So if you want more information on that information I'll give you towards the end of the class. So stay till the very end. Okay. Now, let me ask all of you a simple question about eukaryotic cell. Okay. See, all of you told me, students, entire prokaryotic cell is found in your kingdom Monera. Yes, the entire prokaryotic cell is found in your kingdom Monera. So, which type of kingdoms do you think has eukaryotic cell, students? Anyone in the chat who can tell me? Anyone? Students? Anyone who can tell me, students? Anyone? Students, don't worry about the notes. I will give you all the notes, students. I'll give you all the notes. Nothing to worry. Okay. Students, quickly in the chat. See, prokaryotes, kingdom Monera. Prokaryotes, kingdom Monera, we established. Okay, you're fine. But where do you find eukaryotic cell, students? Let me tell you. The entire eukaryotic cell can be found in your protist, right? Protist or kingdom Protista also. Yes, kingdom Protista. Students, what about kingdom Fungi? Yes or no? You can find them in kingdom Fungi. Okay. We can find them in kingdom Plantae. We can find them in kingdom Animalia. So can I tell except kingdom Monera, everyone is just chilling with eukaryotic cell, students? Except kingdom Monera, everyone is just chilling with your entire eukaryotic cell? Yes, we are, students. That is the right answer. Now, what do you actually find in a eukaryotic cell? Inside a eukaryotic cell, students, do you find nucleus? If yes, tell me in the chat, students. So in a typical eukaryotic cell, we find nucleus, we find nuclear envelope. Moreover, students, we find membrane. bound organelles. So all the organelles here are membrane bound. But students, do you think there are any membranes, any organelle here, two of them, okay, which do not have any membrane? Anyone who can tell me, students? Anyone? See, there are two organelles here. One is called as your entire ribosomes, right? Other one is called as centrosome. Right? Centrosomes. Now these two organs, students, centrosomes and ribosomes, do not have any membrane. Like, they do not have any membrane, students. That have no membrane. So please remember this exception. Sometimes, somewhere, in some way in your life, they might ask you an examination, students. Okay. Now, here, students, in the case of eukaryotic cell, they have something called as cytoskeleton. Okay. Cytoskeleton. So don't worry, students, what is cytoskeleton? I'll teach you what is cytoskeleton. They have complex locomotory structures like your cilia and flagella. I will teach you the entire concept today. Nothing to worry, students. Now, if you are a student like this right now, who thinks only thing they know right now is mitochondria is part of the cell, students, trust me, by the end of today's class, you will not be a SpongeBob. You will know the entire detail about every single organelle from this particular chapter. Okay, students, is everyone ready? Students, all of you ready for the first organelle? The first organelle, before we go, right, there is something called as plant cell and animal cell. Now, all of you know the basic difference. Plant cell has plastids, plant cell has large vacuole, plant cell do not have centrioles. So based on that information, all of you, Anurag, Rohan, Aishwarya, Varshita, based on the information, can anyone tell me which of the following is a plant cell? A or B? Quickly in the chat, students, quickly in the chat. Which one of the following, right? Which one of the following is a plant cell? In the chat, quickly, all of you. Everyone. So you'll obviously tell me, sir, wait a minute, I can find here cell wall. I can find cell wall here. Done. I can find here something called as your, where students? Chloroplast here. Chloroplast here, students. I can see a large central vacuole here. So I will call this plant cell, obviously, students. And if you see here, this is your typical animal cell. No cell wall. There is nothing called as chloroplast. Only thing it has extra is your centriole. Be happy. Okay, animal cell. Okay. Students, we are team plants. All of us are team plant, okay, in my class. Now, students, the first organelle which we'll be talking about here is cell membrane or plasma membrane. Okay. Now, while I tell you about this information about this entire, can anyone tell me who gave the inspiration? Students, anyone? Who do you think gave us the inspiration for the formation of cell membrane? Anyone? Where do you think the inspiration came from? Okay. Let me just rephrase the question. How many of you love drive car, to bike or bicycle? Students, how many of you basically try to go on road? Or how many of you been on the road in a vehicle? Students, should I reveal the secret? Students, should I reveal the secret? Who is the inspiration, the ultimate inspiration, students, ultimate inspiration for cell membrane? Students, it is our Indian roads. Students, it is our Indian roads. Let me show you, students, let me show you. Okay, students, see this. You will tell me, just like our Indian road it is. Just like our Indian roads. Okay. Students, this right here is our, students, this right here is our cell membrane. Students, imagine, let's say Aparjita, okay, he is going, students, he is going, nicely bike is going, dubuk, one hump, suddenly one depression, one full undulation, one more hump. So this is a typical inspiration derived from our roads only. See this, look this, student. This is your cell membrane. This is your typical cell membrane. I can't tell difference only, first of all. I can't tell the difference only. Okay. So this is your typical cell membrane here. Almighty. Okay. Now, students, I want all of you to understand, right? First of all, the cell membrane is living. All jokes apart, cell membrane is living. Okay. It is very living, like all of you. Okay. Cell membrane is living in nature. Now, students, it is also asymmetrical. Students, this is also asymmetrical. right? Asymmetrical. Now, students, all of you, have you ever observed this in your house? See, if you have old people, if you're old people, okay, like if you have old rich houses, like back in the day, see, nowadays everyone puts granites, nice granites they put, nice tiles they put, but this is the OG house. So in all the OG houses, right, in all the OG houses, you will find something like this on the floor. Okay, now students, can anyone find any pattern in this? Students, do you find any pattern here, students? Any pattern here? Anyone? No. You cannot find any pattern. So this entire thing, student, this entire thing is called as mosaic. Mosaic means random. Mosaic means random. And this is how your cell membrane is. The entire cell membrane is completely random. So let me show you. Let me show you. Students, can all of you make any sense out of it? Friends, any sense out of it? Can you see here? It is so random. Suddenly one hump here. Don't ask me why is a donut here. This is not a donut, okay? Not a donut. Okay. There is a hump, there is an aquaporine here, one more protein coming from somewhere, right? So the entire cell membrane is asymmetrical, right? The entire cell membrane, students, it is asymmetrical. It is mosaic or random in nature. It is random in nature. And apart from the students, apart from that, the entire cell membrane, it is also semi-solid. Semi-solid. This semi-solid has a better name actually. This semi-solid is also called as quasi-fluid. Quasi-fluid. Same meaning, same meaning. Now, you'll ask me, sir, what do I mean by quasi-fluid? Okay. Students, how many of you love eating jelly? Raise your hands. How many of you love eating jelly? This one. Student, this one. How many of you love eating jellies? Yes, sir. All of you love eating jelly. It's very tasty and nice, chewy and everything. Your cell membrane is like this jelly only. It is not completely solid. It is not completely liquid. It is semi-solid in nature. It is semi-solid in nature. Why? Because here, this entire jelly, students, let's say I will make one swimming pool. One big swimming pool from jelly only. Can you go jump on that jelly? You can. Okay. So is movement possible in this jelly? Yes or no? Is the movement possible? If I take someone here and I'll give them for swimming, nice, they can move inside. You can move inside. Okay. So you can move inside there. It is called a semi-solid in nature because it has solid-like property, but at the same time, you can move in it. The same property also applies to your plasma membrane. Plasma membrane is semi-solid in nature, quasi-fluid in nature. You can move in it. Okay. Now, students, now let's come back here. Let's come back here, students. Now, all this aside, right? Living, asymmetrical, random, quasi-fluid. What do you think the cell membrane is made up of actually? Nice question, no? Nice question, no? Let me answer, students. What is it made up of? The entire cell membrane is made up of mainly proteins. It is made up of lipids. It is made up of carbohydrates. Carbohydrates, right? It is also made up of water to some extent. A little bit of water here and there. Okay. Now, students, apart from all this, there is something called as a stabilizing agent here. The stabilizing agent is actually a type of lipid which all of you do not like. Some of them you need, but some of you don't like it. That is called as cholesterol. Okay, that is called as, students, cholesterol here. That is called as cholesterol. So what is lipid made of, students? The entire cell membrane is made up of proteins, lipids, carbohydrates, water, cholesterol. It is living, asymmetrical, random, semi-solid in nature. Any doubt till now, students? Any doubt till now, all of you? This is its random. This is your entire cell membrane here. This is how it looks. Now let's take a deeper examination. Like, how does it actually look from inside? Students, the entire cell membrane is made up of two layers of phospholipid. It is made up of two layers of phospholipid. That is why I will call it as lipid bilayer. Students, see this. That's why I will call it as, students. That's why I will call it as lipid bilayer, students. Where did we go? Once again, students. Once again. Students, see this. This will make sense to all of you when we see this. Actually, we see this. Okay. Students, if you focus here, can all of you see here there are lipid molecules here, continuous one layer, phospholipids here. There is one more, students. Cholesterol is a stabilizing agent there. Okay. Now, can all of you see one more layer at the bottom here? So it is a bilayer here, bilayer. So there is a lipid bilayer here, lipid bilayer. Okay. Now, students, apart from lipid bilayer, I told you there is proteins here. Okay. Now, let me ask you a simple question. Okay. Let me just ask you a simple question. I need a paper here. Okay, students. Very simple question. Magic, no, there's no magic trick. Okay, no magic trick. This is a simple paper here. Simple paper. Okay. Now, if I take this pen and I keep it inside this paper, okay, directly inside like this. Tell me, students, is it difficult to remove or easy to remove? It is somewhat inside right now. Difficult to remove, right? Difficult to remove. The pen is very difficult to remove. Okay. Now what about now, students? Tell me. Now, what about tell me now? Easy to remove, difficult to remove now. Now I can hold it easily and I can remove it easily. See, in the first case, it was completely inside. The pen was completely inside. Difficult to remove. In the second case, the pen is almost outside. Very easy to remove. The same concept also applies to proteins here. Based on the ease of extraction. When I say ease of extraction, I mean how easily I can remove them. Now let's say, students, some protein is somewhere in the outside like this, students. Some protein is somewhere outside like this. Now, can I remove this protein easily out? Tell me, yes or no, students? It will be easy to remove. Now, such proteins which are present on the outside or somewhat outside, they are called as peripheral proteins. Peripheral proteins. Now, friends, some proteins, you can see this one, students, this blue one here. Look at this blue one and this green one. This one is completely on the inside. Tell me, students, will it be easy to remove this or difficult to remove this? You will tell me difficult to remove, right? Difficult to remove. So this type of protein is called as integral protein. Integral means, students, integral means it is completely on the inside. Integral or transmembrane protein. Okay. Integral is completely on the inside. Okay. Integral is on the inside. So this and these are the proteins here. These are the proteins here. Proteins. Okay. So let's just come back to the diagram here, students. Let's just come back to the diagram if the stupid thing stops overacting. Now, students, where were we? Now, this is your lipid bilayer. That concept is easy to all of you understood, no? The protein concept. Okay. Now, this is a phospholipid bilayer. Two lipid molecules are here. Okay. Two layers. One layer, second layer here. Two layers. Now, let's focus on one lipid here. One lipid molecule. This is one lipid molecule here. This one lipid molecule, students. This one lipid molecule. Okay. One lipid molecule here has two parts. It has montetale, okay. It has the head region, it has the tail region. The head region is called as polar head region. Okay. See, all of you know basic chemistry, polar, non-polar molecules, all of you know. Yes or no? Right. And this polar region, students, polar head is hydrophilic in nature. Meaning of hydro is water. Philic, loves. So this complete head region, it loves water. It will interact with water. Now this tail region, so tail region is non-polar tail. It is non-polar tail here. Okay. This non-polar tail is hydrophobic. Like, water, 1 kg water. Okay. Sorry, 1 liter water. Who is kg? Okay. Now, students, this is hydrophobic. It means it doesn't like water. Doesn't like water. Okay. So here, motetale here, this person likes water. Hydrophilic, hydrophobic. Hydrophilic, hydrophobic. Okay. Now, they are arranged like this. Okay, they are arranged like this. Okay, students. Now, students, see, this is the entire cell membrane. This is how they are arranged. Can all of you see? This is how they are arranged properly. This is how they are arranged properly. Okay, this is how they are arranged. Okay. Now, students, now we come to NCRE diagram now. NCRE diagram. Okay, NCRE diagram right now. Okay, students, can all of you see the lipid bilayer here? One layer here, second layer here. Lipid bilayer. Okay. It has protein on the inside called as integral protein. This protein is very difficult to remove. Difficult to remove. Very difficult to remove. Okay, I'll tell you, students. I'll tell you. Passion, graduation, and coach cooking. I'll tell you 1 second. This integral protein is very difficult to remove. Students, do you see one more protein here? Protein on the outside here, students. This is called as peripheral protein here. Peripheral protein is protein on the outside. They can just hold and remove it out. Okay, very easy to remove, difficult to remove. There is cholesterol, students. Cholesterol is your stabilizing agent here. Okay. Then we see sugar, students. What is sugar? Sugar is nothing but carbohydrate. Sugar is nothing but carbohydrate. Okay. Then we have lipid bilayer, students. Lipid bilayer. Now, students, the entire diagram is decoded. Yes or no? Now, this actually is a model, not the model from the TV and all. This is a different model. This model is called as fluid mosaic model. Students, it's called as fluid mosaic model. Now, this model's namkarana, okay, naming ceremony. Naming ceremony of this entire thing was given by Singer and Nicholson. Singer and Nicholson in 1972. 1972. Singer and Nicholson, students. Singer and Nicholson. Now, sugar means carbohydrate. Glucose is a type of carbohydrate. Okay. Glucose is a type of carbohydrate. Sugar means basically carbohydrate. Okay. Now, this is called as fluid mosaic model. Students, why is it called as fluid here? Anyone? It is called as fluid because like a jelly-like structure, things can float in it. It is called as mosaic. Can anyone tell me why is it called as mosaic? Anyone, students? Anyone? Because it is called as mosaic because entire thing is very random. So, students, does it make sense now? Fluid mosaic model. It is fluid in nature. It is completely random model given by Singer and Nicholson in 1972. Thumbs up in the chat, all of you. Now, I told you two proteins. One is transmembrane or integral protein on the inside here. And we have the peripheral or extrinsic proteins on the outside here. Outside the house, inside the house. Simple as it gets, students. Simple as it gets. Okay. Now, students, I told you all of you, we can find movement here. Actually, students, we can find two types of movements here. We can find two types of movements. One is called what, students? One is called as flip-flop. Flip-flop movement. Okay. Other one is called as your lateral movement. Lateral movement. Students, very simple concept. Listen to me, all of you. Very simple concept, students. Yes, they are scientists. Scientist name only Singer. Imagine. You can keep whatever you want. Okay. Now, students, I'll teach you what is flip-flop first of all. Just listen to me. Very easy it is. NCR doesn't teach you very well. You're in my class, you learn and go. Learn everything in the class only. So this is your lipid bilayer, students. Lipid bilayer. What do I mean by flip-flop movement, students? What do I mean by flip-flop? This will go here. This will go here. Basically, it is exchange offer happening. This person, one person will go here. This one person will come down. That is called flip-flop movement. Flip-flop movement. That is called as flip-flop. Okay. A lipid can undergo. They will change the membrane only. They'll from lower. They'll go to upper to lower. Simple as that. Flip-flop. Okay. Now, what is lateral movement, students? I'll draw it here only. You understand. Okay. What do you mean by lateral movement? Understand, students. Here inside I have proteins here. Let's say I have protein on the inside. Let's say I have a protein on the inside here. Let me just paint this protein here. Okay. I have protein on the inside right now, students. I have a protein on the inside here. Okay. This right here is a protein. This right here is a protein. Okay. Here is your protein. Okay. Now this protein right here, students, this protein right here, it will undergo lateral movement. How? Let's say I'm a protein here. I'm a protein here, students. Okay, I'm a protein. I will go move like this. I'll go move like this. I'll go out of the frame. Okay. So this protein here can show lateral movement. Protein can show what? Lateral movement. While your lipids, students, lipids, flip-flop movement. Flip-flop movement can be shown by lipids only, while your lateral movement can be shown by proteins and lipids. Both can show lateral movement. So this is called lateral. So if I'm here, I'll go like this. Lateral. That is called as lateral movement. This is called flip-flop movement. Is that point clear to all of you students? Students, is that clear to all of you? Mr. Passion, gardening and cooking, I'm coming there only now only. Okay. Students, is this concept clear to all of you? If this concept is clear to all of you, drink some water, all of you. Drink some water quickly. All of you drink some water. Done. One concept at a time. Now students, now. That is a movement. Students, that's why I said cell membrane as what? Cell membrane as what? Semi-solid, because we can find movement there. Movement can be seen here. Okay. Now students, what do you think is the function? Students, what do you think is the function of cell membrane? Anyone? What do you think is the function? Pushing substance out of the cell or put substance into the cell? Students, what do you think is the function of cell membrane? It is actually both. Okay. So movement of proteins can be a function. Endocytosis, cell division, secretion, formation of intercellular junctions. So all of these becomes the function of your plasma membrane, students. Plasma membrane. So all of these are functions here. Okay, all of these are functions. Okay. Now students, there is something called as movement, right? There is something called as movement, right? Movement. Now here movement can be of two types mainly. One is called as your passive transport. One is called as your active transport. Not activa, active transport. Say active transport. Okay. Students, I should think activa gadi before. Okay. Active transport. Listen to me. Passive transport. Students, how many of you have been to Tirupati? Raise your hands. Or how many of you have gone and stood in front of a bus? You're standing in a bus stand. Imagine there's a bus coming. Woo, bus came. You're standing near a bus stand. Okay, bus stand, you're standing. Bus came. Empty bus came. Okay. Tell me students, where do you have more people? Empty bus is coming, right? No one is inside the bus. Large crowd outside the bus. Now students, what do you need to do? All you need to do is go stand near the bus, right? Automatically, people will push you. Push. From outside more people to inside the bus, more people will become now. See students, bus is coming, you're standing there, automatically you're going and standing inside now. That is your passive transport. Okay. There is no energy required here. You will automatically move inside the bus. Okay. Along the concentration gradient, that is from high concentration, that is more people on the outside to the less people on the inside. So high to low concentration it happens. That is called as your passive transport. Doesn't require energy. Automatically you'll be inside the bus. Okay. Now, same concept also applies in your osmosis also. In osmosis. Osmosis happens because of simple diffusion. In osmosis, water is going from high concentration to low concentration. Okay, high to low concentration you go. Okay, that is what happens in the case of passive transport. No ATP required. Along the concentration gradient, high concentration to low concentration. Done. Now students, what is active transport? Now students, as the name says, active transport means it requires energy. Okay. So what happens generally students? All of these polar molecules, right? Stupid polar molecules. Okay. Polar molecules, there is no entry. Usually what happens students, in this lipid bilayer, right? Polar molecules cannot enter inside. Not enter inside. So they will call a friend. They will phone a friend here. Okay. So all the polar molecules which cannot enter inside, they'll call a friend here. Who is the friend students? The friend here is carrier protein. Okay. So he'll call a friend, bro, I'm not able to go inside. You should take me inside now. The bro is like, don't worry bro, I got you. Okay. So carrier protein will catch hold of your polar molecule, use the ATP and enter from where? And enter from low concentration to high concentration. That is against the gradient. Against the gradient it goes. Okay. So what is polar molecule students? Do not worry, you will learn all that in your chemistry. See, if I teach you basic, it will not make sense right now. So if you want to understand what is polar molecule, non-polar molecule, chemistry, best the Adiksha ma'am will teach you. Okay. Just remember students, polar molecule charge wise different from your non-polar, right? Usually polar molecules cannot enter the membrane here because of the hydrophilic. Positive and negative charge. Yes. Okay. Now students, is this clear to all of you? Polar molecules cannot enter inside. Carrier protein can enter with the help of carrier protein, use the energy and they can go from low concentration to high concentration. One of the few examples is your sodium potassium pump is one of the examples. It's a channel there. Okay. Now all this aside, those of you know what is a polar molecule, tell me, is water a polar molecule, students? Anyone can tell me in the chat? Oops. Students, do you think water is a polar molecule? Water, Tanni, Pani, Nero. Do you think water is a polar molecule, students? Is water a polar molecule? Yes, water is a polar molecule. Then how do you think water can enter inside? Water is a polar molecule, students. Water is a polar molecule. But water undergoes passive transport. How? See, I told you just now, polar molecules cannot enter inside. And you also know water is a polar molecule. Then how do you think water undergoes passive transport, that is a diffusion, in diffusion, students? All of you. What is a polygon? I don't even know what is a polygon. Students, how do you think water enters inside? I don't know, sir. I don't know. Mohammed, I'm not sure. Okay. So tell me in the chat quickly. How do you think water can enter inside? How is it possible? Simple answer. Students, simple answer. They have a red carpet for them. Students. Red carpet for them. Students, there is something called as aquaporins. With the help of these aquaporins, aquaporin is a type of your protein molecule here. With the help of these aquaporins, water can easily enter or exit. Okay, and moreover, water is very tiny. Water is so tiny, it can easily squeeze inside. Usually, water is so tiny, students. Water is so tiny, it can easily squeeze inside also. Okay, easily inside. Okay, so water is a polar molecule. It can enter inside, students. Enter inside. So, any doubt, students? Any doubt in the first part of the topic today? Cell membrane, students. Oh, multiple language. Oh, sorry. Students, I know here and there, students, Tamil here, I watch Tamil, I listen to Tamil songs. Telugu movies, obviously I watch. Kannada is my mother tongue. Okay. Hindi because I was born in North India, so Hindi I know. English because of the language. Malayalam because of the friends here and there. So if you're in South India, you end up knowing around 3 to 4 languages here and there. If you cannot speak also, you can understand some of them. Okay. Now students, that is your first part of the chapter is over. So if everything is clear till now, can I get a fire emoji in the chat, students? Can I get a fire emoji in the chat, all of you? Can I get a fire emoji in the chat for all of you? Quickly in the chat. All of you. Fire emoji in the chat. Shh. Students, my dog is acting up today. I don't know why. Done, students? Done? Okay then. Students, now we'll do about your cell wall now. Cell wall is a little different here. Okay. Cell wall is obviously non-living in nature. Papa. Right? Non-living in nature. Protective in nature because it provides protection here. Protection here, students. Outer covering to the plasma membrane. It is a outer covering to the plasma membrane in the case of a plant cell. Students, see this. If this is your plasma membrane, the outer covering is called a cell wall in the case of plant cell. In the case of plant cell, it is outer covering, students. Outer covering there. Okay. Function, students, it is found in your plant cell, fungi, and some protists. It is found there. Functions, shape and rigidity to the cell. Protection, obviously, barrier, right? Prevents from cell from bursting. It will prevent the cell from going blasting. Cell to cell interaction. This barely they ask question here, but I'll tell you what they will ask question mainly, students. Let me just teach you that main part. Okay, this is the main part here, students. This is the main part called as your layers of cell. Okay, now let's say I have a cell here, students. I have a cell here. Let me just draw a cell. Where is the color? Okay, let's say I have a cell here, student. This is your cell wall. Okay, here we have the oops. This is the plasma membrane right now. Okay, this is called as primary cell wall. Okay, this is the plasma membrane here. Okay, okay. Now students, here this primary cell wall is present in your young plants. It is present in the young plants. Okay, it is much more permeable and flexible. Okay, it is present in your young plants. It is much more permeable and flexible. Okay, flexible. Okay, it will allow things inside. Now students, let me ask all of you a simple question. This primary cell wall will diminish, RIP. It'll go away some other place. Okay, diminishes. Now after this, there is a birth of secondary cell wall. Now students, you are supposed to tell me, where do you think we find secondary cell wall? This is primary cell wall here. This right here is the primary cell wall here. Where do you think we can secondary cell wall? Do we get secondary cell wall on the outside like this, or here? Where do you think we get the secondary cell wall, students? Do you think we get the secondary cell wall on the outside or the inside? All of you. Students, where do you think? Where do you think we get the secondary cell wall? All of you. Quickly in the chat, students. Quickly in the chat. Students, listen to me very carefully. If this is your primary cell wall, students, the secondary cell wall will arise on the outside here. On the outside, we will get the secondary. Outside, students, let me quickly in the chat. Outside or inside? Students, quickly in the chat. Outside or inside? Students, where do you see now? Tell me. Tell me, students, outside or inside? No, tell me. At least now you tell me. Quickly in the chat, students. Is it outside or inside? I hope it is very clearly visible. All of it is visible now. Tell me. On the inside, we get the secondary cell wall. Okay. Now, students, secondary cell wall, very rigid. It is very rigid, not permeable at all. Not permeable at all. Okay, very rigid, very very, it becomes like how all of you in the primary you were all sweet, but as soon as you come to secondary, as soon as you hit the puberty, all crankiness, all mood swings, same thing. Secondary cell wall is very rigid. It not permeable at all, not flexible. Just want to stay there, not do anything. Okay, that is the secondary cell wall on the inside, students. Secondary cell wall on the inside. Okay, now students, let me just draw one more here. Let's say one more cell is here. Let's say one more cell is here. This is one more cell next to it. This is a neighboring cell. This is the neighboring cell, students. Now between these two cells, this middle part is here, right? This middle part is called as middle lamella. This entire middle region is called as middle lamella here. This entire region is called as middle lamella here. This is called as middle lamella. Middle lamella. Now, sometimes they can ask you, students, what is it made up of? The middle lamella is made up of calcium pectate and magnesium pectate. It is made up of calcium pectate and magnesium pectate. Okay, so this right here, students, Okay, this right here, right, is called as your middle lamella, right in the middle here. This is the primary cell wall, secondary cell wall, right, students? Hello, Ravindran students. Hello, Ravindrani. How are you? Right. This is the middle lamella here, and primary cell wall, secondary cell wall. Okay, now students. Now, what is it made up of? What is the cell wall made up of? The cell wall is made up of multiple different things in different organisms. In the case of algae, it is made up of cellulose, galactans, mannans, calcium carbonate. At the same time, students, plant cell, it is made up of cellulose plus hemicellulose and pectins. In the case of fungi, it is made up of chitin. It is made up of chitin in the case of fungi. That is your entire cell wall, students. That is your entire cell wall. Exactly. The outer one gets destroyed and we have the inner one on the inside. Okay. Now, students, now, can all of you see, students, here? This is called as middle lamellae right now. Middle lamellae here. Now, students, I want all of you to understand here this particular, right? Here we have the cytoplasm here. Here we have the inside cytoplasm, and here we have the cytoplasm. Okay. Now, students, I want all of you to understand the cytoplasm, right? This cytoplasm is actually connected. This cytoplasm, students, it is connected to the adjacent cytoplasm here. Can all of you see, students? The cytoplasm is connected here. This is called as cytoplasmic connection, or it is called as what? It is called as plasmodesmata. Students, can you see here? It is called as cytoplasmic connection. It is called as what? Cytoplasmic connection. Cytoplasmic connection or plasmodesmata here. The cytoplasm of this and cytoplasm of this are connected. They're called as plasmodesmata. As students, remember, this plasmodesmata is present in your young dividing cells a lot. Young dividing cells. Okay. It is present a lot. Okay. That is your plasmodesmata right now. So, plasmodesmata, cytoplasmic connection, same thing. Here we have middle lamellae here, students. Here we have the middle lamellae here, Then we have primary cell wall, secondary cell wall on the inside. Yes. Then, students, all of us know what is it made up of. Then we know cell wall is non-living, rigid, outer covering of the plasma membrane, plant cell, fungi, and some protists we can find them. With that, your entire cell wall is over. Done. Done, students. Done. Okay. Now, students, now we'll start something called as your endomembrane system. Ready, all of you students? Ready? Shall we start, students? Shall we start? Before we start, all of you drink some water. See, water is very important. All of you drink some water. Protist is basically a kingdom. Protist is a kingdom, no? You didn't attend ma'am's class, Gopika ma'am's class, biology classification. In biology classification, we learn, right, protist, protista. We learned about your plant kingdom, animal kingdom, right, Monera, then we learned fungi. You learned, right, all of you learned, right? Okay. Now, students, in endomembrane system is actually like a gang. Students, in your school, all of you have gangs, right? In your school, all of you have gang, like my gang, your gang, opponent gang, your friend's gang. Now, students, in your class or in your friend circle, when you try to make a gang or a team or a members, do you choose people based on the similarities, based on the interest, or any random or any random people? Or any random people, students? All of you? All of you? See, in endomembrane system also, students, there are here 4 organelles. They're best friends. They talk to each other. Okay, their functions are coordinated here. Okay, now to remember this, there's a small trick here. The small trick is, students, every gamer loves victory. Every gamer loves victory, students. Students, all of you play games? Students, how many of you play games? All of you? How many of you play games? See, E for endoplasmic reticulum. Endoplasmic reticulum. G for Golgi complex or Golgi apparatus. Golgi complex or Golgi apparatus. L for lysosomes. L for lysosome. V for vacuole. V for vacuole here. So all of these students, all of these four organelles here, all of these four organelles, they are part of endomembrane system. Why? Because their functions are coordinated. Because their functions are coordinated. Okay. Now, students, here is a quick question to all of you students. Okay. Students, if you want to know about all of them, are you ready? Okay. Because, yes, see, students, I love memes. I love memes to the core. I will show you one meme right now. Let's see how many of you understand, students. How many of you understand this meme? See, you will understand this meme only if you have the knowledge. Okay. So I will trust me, students. Trust me. If you do not know what is a lysosome, right? If you do not know what is a lysosome, you will not understand that. Okay. So next 25 minutes. In next 25 minutes, I will teach you every single organelle. Then we'll come back to this meme and you will say like, this is very funny. Then you will say very funny. Okay. So right now you might not understand because you might not have read the chapter. Now I will teach you one by one and then we'll see how it is. Okay. All of you students, all of you ready? Okay. Now before this, one small question. Can I get the answer? Can I get the answer quickly, students? Can I get the answer quickly? The membrane-bound organelles of endomembrane system coordinate cellular functions. True. Mitochondria and chloroplasts are not a part of endomembrane system. True. They are not a part of endomembrane system. Okay. Students, the organelles that are included in the endomembrane system are the organelles which are part of your endomembrane system. See, mitochondria is not a part of it. Mitochondria not a part of it. Mitochondria not a part of it. Last option. Last option, students. Okay. Now the first organelle here is called as endoplasmic reticulum. It sounds very difficult, but it's very easy. It's a pavam person. Okay, very easy, very what do I say? Very simple person. Okay, endoplasmic reticulum. The first gang member, students. The first gang member is called as your endoplasmic reticulum, students. Okay. Now endoplasmic reticulum is nothing but try to imagine one round structure like a cell. In that we have a lot of straws, lot of pipes, lots and lots of pipes everywhere, students, lots and lots of pipes everywhere. That is ER. That is, students, is ER. So after the electron microscope, right, invention, they found that right now there is a large network or reticulum of tiny tubular structures throughout the cell. That is called as your endoplasmic reticulum. Students. Now this endoplasmic reticulum, students, it is actually extension of nuclear membrane. From the outside of outer nuclear membrane, we can see it as an extension right now. Okay, let me just show you, students. Okay, let me just draw a cell here. This is a cell right now. Students. This is a cell. In this cell, students, there is a brain. All of you know the brain, right? Not the brain rot, different brain here. This is the brain of a cell called as nucleus. Now nucleus has two membranes, outer membrane and inner membrane. Okay, let's say this is the inner membrane here and outer membrane here. Okay, inner membrane and outer membrane here. Okay, now students, from this outer membrane right now, we can see some tubular structures. Tubular structure like this. From here also we can find tubular structures arising from the outer layer. Extension of nuclear membrane, exactly. From the outer layer of nuclear membrane, we can see some tubular structures arising. Now, these tubular structures are called as your ER or endoplasmic reticulum. Endoplasmic reticulum, students. Endoplasmic reticulum, right now, students. Endoplasmic reticulum. Now, students, this entire endoplasmic reticulum, it will divide the entire cell into two parts. Okay. Now, we see this entire cytoplasm, right? This entire thing is called as cytoplasm here. This entire thing is called as cytoplasm. Okay. Now, students, in the cell, inside a cell, wherever we find the ER, this green region, wherever we find the ER, the green region, this region is called as luminal region. Luminal region. Okay. And wherever we do not find, in some places we do not find your ER, right? This region is called as extra luminal. So luminal and extra luminal, students. Okay. So the ER, the entire ER is dividing the cell into two parts. Entire cell into two parts. Wherever we find ER, right? ER, that part is called as luminal part. Wherever we find the cytoplasm here, this entire region is called as extraluminal, students. So luminal and extraluminal are the two parts of a cell when we see in accordance to the endoplasmic reticulum. So it divides the entire cell. It divides the entire cell into two parts, luminal part and extraluminal part. Is that point clear to all of you, students? Is that point clear to all of you? Very simple it is. Okay. Now, students, the ER can actually be of two types. Okay. If ER has something called as your ribosomes, if it has ribosomes in it, then it is called as RER, rough endoplasmic reticulum. Then it is called as rough endoplasmic reticulum. Okay. Now, students, let's say if the ER has no ribosomes, it has no ribosomes, it is very smooth. Okay. In such a case, it is called as SER, smooth endoplasmic reticulum. It is called as smooth endoplasmic reticulum, students. Okay. Now, students, function. This RER main function is protein synthesis. Protein synthesis. Why? Because rough endoplasmic reticulum, students, rough endoplasmic reticulum has ribosomes. That is why the main function is protein synthesis. Now, what about RER, students? SER. The SER main function is lipid and students, lipid as well as your the board is too low. I'm not able to bend right now. I'm trying to like crouch and hormone synthesis. And hormone synthesis. Now you ask me, which type of hormone? It produces steroidal hormones. Can anyone give me the example of steroidal hormones in the chat? How many of you know? Example of steroidal hormones. Anyone? Students. Steroidal hormones. Anyone? Example of steroidal hormones. Testosterone, estrogen, progesterone. Yes. All of them are called as your steroidal hormones. Okay, steroidal hormones, students. Now, students, how many of you learned human physiology? How many of you learned about human physiology, students? In human physiology, when you learn something called as muscle contraction, do we see ER there to release an uptake of calcium? Yes, we do. So the one more function of ER, students, one more function of ER is muscle contraction. It also helps in muscle contraction. Okay, muscle contraction. Now, students, apart from Apart from muscle contraction, it is also involved, students. It is also involved in other functions. It is also involved in other functions also. Okay, there are a lot of other functions, but these are main functions here. For example, detoxification of drugs. Okay, detoxification of drugs also. I'll just write down if you want. Detoxification of drugs also. Okay. Again, students, if you are new to NEET examination, you will learn one by one every single chapter. What is steroidal hormones? You learn in chemical coordination. Okay. You learn about muscle contraction in your locomotion chapter. Okay. There, Govika will teach you how does the muscle contraction happens and how does the calcium plays an important role in muscle contraction, one by one. Okay. So there, microscope, after electron microscope, network of reticulum of tiny tubular structures here. Can you see? Network of tubular structures. Okay. Now again, students, if you want extra information, this entire ER can be in the form of vesicles also. It can be in the form of vesicles. It can be tubular. It can be cisternae also. Okay. There are multiple other forms depending upon where are they found. Let's say in the case of liver cell, and pancreas, you will find more ER. Why? Because they are metabolically active. In the case of adipose tissue, you will find tubular ones, right? So similarly, in the case of spermatocyte, we can find the vesicle form. So there are multiple other information. I just stick to this much NCERT level right now. अगर मिरर वेज होता तो आप उसे क्या बोलते? I don't know, students. If mirror is veg, minor is veg. I don't know, students. Okay. Now, students, when I told you it is a network of reticulate of tiny tubes, this is how it looks in electron microscope. Students, all of you can see? Students, all of you can see, right? Tell me yes or no. Students, does it make sense to all of you now? Does it make sense to all of you, students? Yes or no? All of you. See, here we have the nucleus here. Nucleus. Multiple tube-like structures. Can you see? Multiple tube-like structures. You can find some vesicle forms somewhere known. Ribosomes everywhere. Throughout this, you can see small dots here. Those are the ribosomes here. Okay. This is the entire network inside a cell. This entire part is called as luminal. Luminal part. See, this is the cisternae part here. Okay. Extension of nuclear membrane. Okay. See, students, here. This is our extension of membrane. Okay. Now, students, that's how it is. One question to all of you, students. Quick answer. I want, students. Quick answer. Which of the following statements concerning the endoplasmic reticulum is incorrect? See, ACR is a site for lipid synthesis, is correct. Right? RER has ribosomes. SCR is devoid of ribosomes, yes. Wrong answer. Okay, incorrect. Because in prokaryotes, we do not have anything. Okay. Students, next one is called as Golgi complex. Now, first of all, who discovered Golgi complex? All very basic, called as Camillo Golgi. Okay. Camillo Golgi discovered the Golgi complex. He found a densely stained structure. Near the nucleus, students. Okay, and he found them, all of them very flat. They were flat disc-shaped structures. All of them were arranged parallelly to each other. Students. Okay, arranged parallel to each other. Let me give you a perspective how they look like. Okay, now, students, this is a nucleus, let's say, and this is an entire cell. This is an entire cell. Here we have nucleus. Nucleus has two membrane. Nucleus has two membranes here. Okay. Now students, from this outer membrane, what do we get? Anyone? From this outer membrane, we get ER. Here we get the ER. Okay. Now students, just near the ER, here we can find certain concentrically arranged structures like this. You see students, here we can find certain concentrically arranged structures just near the nucleus. These are called as your Golgi complex. These are called as the Golgi apparatus or Golgi complex. Now students, I want maximum attention for the next 5 minutes. Can I get maximum attention for next 5 minutes? Maximum attention, because what I'll be teaching you right now is very important. What I'll be teaching you right now is very important. Okay. Now students, let's say this entire Golgi complex has two faces. You might have met some people, no, in your life. In front of you, they're somehow, behind your back, they're like, ooh, it's bad people. You might have seen, right, some people who say bad things behind your back. Golgi body is like that. Golgi body has two main faces here. Okay. Now students, here, this face here, this one here, this face is called as your cis face, right? Cis face or forming face, or also called as convex face. Any one name is fine here, right? All of you. Okay. Cis face or in fast. Okay. Now this face is called as what, students? This face is called as cis face, right? Cis face, forming face or convex face. Okay, right? Convex face. This part here. This part. Okay. Now it has one more face here. This part. This face is called as what? It is called as trans or maturing or concave face. Okay, concave face. Now you might be wondering, all of you might be wondering, right? Now all of you might be wondering, why did I tell you about two faces here? What does it have to do? It has to do with the function of Golgi complex. The main function of Golgi complex is like Amazon factory. The whole point of Golgi complex, it does packaging. Students, all of you order from Amazon. Friends, tell me, how many of you order from Amazon, students? How many of you order from Amazon? See, if you order from Amazon, Flipkart or Miso, whatever you call it, all AliExpress, okay, when the delivery is coming to your house, it is nicely packed around it, packaging. Inside your cell also, this particular organelle called as Golgi complex does the packaging work. Okay, here let's say some form of molecule is entering here. It could be a protein or it could be a lipid, anything. Okay, it undergoes packaging, and it is exiting out of a packed form. Right? Exiting is always trans phase. Entry is always cis phase. Entry, students, remember, cis phase is always towards the nucleus. Trans phase is always towards cytoplasm. Always towards cytoplasm here. Can you see? Always towards cytoplasm, this one. This one is towards the nucleus. So it enters here, enter, packaging, exit out of trans phase. That is the main, students. That is the main function of Golgi complex, is packaging. Students, here, this is your Camellia Golgi, right? Camellia Golgi apparatus near the nucleus. Okay. Students, see here. Here we have the cis phase. Then this right here is the trans phase. Trans phase here, students. Okay. Cis phase, it is entering, undergoing packaging, and exiting from trans phase. Cis phase is also called as forming phase or convex phase, while this is called as maturing or trans or your concave phase. That is the main functioning of your Golgi complex. Okay, exactly. Now students, see here also. See, protein is entering here. Protein is entering here. It undergoes packaging. Protein is packed, exiting out. This here is the cis phase, entering near the nucleus here. Exit is happening from trans. Exit is happening from the trans phase here, students. That is the concept of your Golgi complex, students. How many of you understood? Raise your hands or put fire emoji in the chat or put any emoji in the chat. Put any emoji in chat, students. Because when you understand the function, because students, all of you need to understand, Golgi complex will always produce something called as glycoproteins and it will produce something called as glycolipids. Okay, this protein part came from RER. This lipid part came from SER. All the glyco part that came from Golgi complex. That is a carbohydrate. That is the carbohydrate here. So the whole point of your Golgi complex is protein from your RER, lipid from SER. All it does is packaging. It will add one. It will take one it will take one carbohydrate. Pachak, paste, next, paste, next, paste, next. That is the main function of Golgi complex. That is the main function of Golgi complex here. Okay, students. Apart from this, all of you know there is something called as your acrosome. All of you know acrosome. Acrosome also formation also happens because of Golgi only. Because of Golgi only. Acrosome. All the enzymes present there. All the enzymes present there because of Golgi complex only. There is something called a cell plate formation because of Golgi complex only. Okay. All it is because of Golgi complex itself. Students. Okay. Now students, in Golgi complex, I want all of you to understand, students, this cis phase and trans phase, they are not identical to each other, but they're students. Students, do you think they're identical to each other? All of you. They're not identical, but they're interconnected. They are not identical to each other, but they're interconnected, students. Interconnected. Students, function of acrosome. Acrosome helps during the when the sperm comes in contact with the egg. There are multiple layers on the egg. All of you know there is something called as zona pellucida, corticorno radiata. All that entire thing happens. Degradation happens because of your this thing only. Okay, students, what is the answer? What is the answer, students? See, their Golgi complex has arranged rear nucleus, right? We have convex cis or forming phase, concave trans or maturing phase. Cis and trans phase, the organelles are identical to each other? Nope. Are they interconnected? Yes. Are they are interconnected? Yes. Are they identical? They're not identical. They're not identical. Okay. Okay, students. Done. Then students. Next question. Next question. All of you. Quick, students, quick. All of you, quick. They are not identical to each other. They are not identical. They are just interconnected to each other. Okay. Students, next person of the gang is called as lysosomes. Next person is called as lysosomes. See, Golgi bodies again. I'm telling you, two faces. Are the two faces same? No. One is front face. One is behind your back. Front of you, they are very, very happy. They are like, very good. They'll praise you. But behind your back, they'll scold you. Okay, two faces, they're not same, but they're connected. How? Because both belong to same person. Students, you can remember it like this. Two faces of a person. Front of you, they're somehow. Behind your back, they are somehow. Okay. Are they same? No, they're not same. But are they connected to each other? Yes, because they belong to the same person. They're connected to each other. Okay. Next person is called as lysosomes, students. Lysosomes. Okay, also called as suicidal bags here. I'll tell you why. I'll tell you why, students. Okay. Now, first of all, lysosomes are actually small, round vesicles. Okay. They're round vesicles. How are they formed, students? Lysosomes are formed by budding of vesicle from your Golgi body. Let me show you, students. Let me show you. See this. This is your nucleus here. Here we have the endoplasmic reticulum here. Endoplasmic reticulum is forming a protein here, students. Protein. The protein is entering the structure here. These are the cisternae here. Right. Now, students, can anyone tell me this phase, which phase is this? Is it cis phase or trans phase? The entry is always from the cis phase. The entry is always from the cis phase, exiting from the trans phase. Okay. So protein produced here undergoes packaging, and exit is happening here. And this is how the lysosomes comes into picture. Lysosomes are, students, what? Vesicles are these structures. These are called as vesicles. Round vesicles. Okay. So entirely speaking, lysosomes are formed by budding off from the where? From the entire Golgi complex. From Golgi complex, they bud out, they come like this. Okay, that is called as the vesicles. Those are called as the vesicles. Can you see here also? See, Golgi apparatus here. Golgi apparatus concentrically arranged. This is called as electron microscope. Okay, cis phase, trans phase, lysosomes. Large lysosomes can be found here from here. They can be produced here. Packaging, they're coming out. Okay, lysosomes are coming out, students. Now, I want all of you to understand all of these lysosomes, right? If anything enters inside, let's say a virus enters inside, it'll kill the virus. Let's say a bacteria enters inside, it'll kill the bacteria. It is basically like your WBC cells, right? It takes care of a cell basically. Now, what if inside a cell, let's say one organelle is not working properly? Do you think it'll kill the organelle also, students? Students, let's say one organelle is not working properly. The organelle is very old. Organelle is like, please. Do you think what lysosome will do? Lysosome will go, it'll kill that organelle also. Now, let's say the cell itself is very old now. Let's say the entire cell itself is very old. In that case, it will kill the entire cell itself. That is called as suicidal bag, because lysosome itself is part of a cell. Lysosome itself is part of a cell only. It is killing its entire cell because it is old, or is it infected? So if it becomes infected, or if it becomes old, it will kill itself. It will kill itself. Okay. So now you might ask me, sir, how do they do it? Friends, how do you think they do it? Do you think it has some special weapon like one shotgun it has, or rifle, or a bazooka? Friends, what do you think your lysosomes has? Friends, here I told you, right? A lysosomes will have some kind of protein. And all of you know, enzymes are basically proteins. They are proteins. So here, lysosomes, students, all these lysosomes, they have something called as, right, hydrolytic enzymes. They have something called as, students, hydrolytic enzymes. And these hydrolytic enzymes are like your hydrolase, lipase, proteases, carbohydrates. All of these enzymes are basically like weapons. They're weapons of mass destruction. They'll go and kill everything. This will go kill the proteins, kill the carbohydrates, kill the lipids, right? Go killing only, killing spree, spread, right? Now, students, there is one condition here. All of these enzymes to work, they need acidic medium. Okay, so all of these enzymes will work at optimum pH, that is acidic medium. So if the acidic medium goes away, they might not function also. They might not function. Okay, friends, let's see how many of you can understand this meme right now. Friends, how many of you understand this meme right now? Tell me in the chat. I found this very funny. I could not resist bringing it here. I read this meme. I started laughing for 10 minutes straight. I'm like, see, something enters toxic enters the cell. Lysosomes. Who's there? I have enzymes and I'm not afraid to destroy myself. Destroy myself, it seems. Oh God. Okay, students. So here is a question to all of you. Let's see who can solve this question. And students, do you realize this meme right now? This one, does it make sense right now? Students, does this make sense right now? Right? Virus or bacteria enters the cell, lysosomes, kaboom, direct, blasting the entire cell only. Blasting the entire cell only. This is what lysosome does, stupid lysosome. Also good also, but you know, lysosomes. Okay, students, question quickly. If the pH of the lysosome is increased to alkaline, what will be the outcome, students? Hydrolytic enzymes become inactive. They'll become inactive because they need, right? Are enzymes lysosomes? Say no. Lysosomes are type of enzymes. Lysosomes are lysosomes is an organelle. Lysosome is an organelle here. Okay. Then we have enzymes separately. Lysosome is an organelle. That organelle has enzymes in it. Okay. It has organelle in it. They're not the same. Quick, students. All of these are NEET PYQs also. Which of the following statement is not correct, students? Lysosomes are formed by the process of packaging of ER. Wrong statement. Packaging of Golgi complex. 1. Packaging of Golgi complex, students. Okay. Now, students, now we come to the last member. Students, now we come to the last member of the group. The last member of the group is called as vacuole. Last member is called as vacuole, students. Vacuole is like a dustbin here. Students, vacuole is like a dustbin. Vacuole is like a dustbin. Students, in your house, right, whatever you don't need, you throw in dustbin only. So inside a plant cell, inside a plant or animal cell, if something is not required, where do you think it goes? Students, inside a plant cell, if something is not required to the cell, some excretory waste, some water sap, some type of anything, everything is thrown inside the vacuole here. Okay, vacuole. Okay, it is a membrane bound organelle space here, students. In the cytoplasm, it contains water sap, excretory products, everything. Here it has a single membrane here. Very important here, very important. The single membrane of your vacuole is called as a tonoplast. I want all of you to highlight this very much. Called as tonoplast. So tonoplast is the membrane. Tonoplast is the membrane of your entire vacuole, students. And moreover, students, in the case of plant cell, 90% of volume is taken by cell only, the vacuole only. 90% of the volume of the cell is taken by vacuole. You can see, students. I'll just show you, students. I'll just show you. Just look at a plant cell here. You'll be shocked. Students, is it visible to all of you? Is the board visible to all of you? Students, is the board visible to all of you? Yes or no? See here, students. This entire cell here, it has your nucleus here. Nucleus is here. Then we have your ER here, just to nucleus. We have ER here. Then we have Golgi somewhere. Golgi. We can have Golgi here also. Now, right in the middle, students, 90% of space. 90% of space is taken by this large, gigantic dustbin here. Okay. The large, gigantic dustbin here is called as vacuole here. This is called as vacuole. Large, gigantic dustbin called as vacuole here, students. Vacuole. Okay. It takes up all the space. Okay. So not only that, this vacuole is very special because this vacuole, it is a dustbin which will take anything. There is no separation here. It will not be like I want wet waste, I only want dry waste, nothing like that. This dustbin will allow all the ions inside. Students, this dustbin here, this stupid dustbin here will allow all the ions. It will allow all the ions to enter inside. It will allow all the ions to enter inside. That too from low concentration to high concentration. No value only. Okay. So from low concentration to high concentration, that is against the gradient, it will allow all the ions inside. Okay. It may look cute, students, as a dustbin. Okay. That is your vacuole, student. That is your vacuole. Okay, students. Now, depending upon the function of this dustbin, depending upon the function of vacuole, there are multiple types of vacuole. There is sap vacuole in the case of plants. In the case of students, in the case of your amoeba, there is something called as contractile vacuole. In the case of amoeba, there is something called as contractile vacuole. Now you'll ask me, sir, what is the function here, students? The function is osmoregulation. Osmoregulation, right? As well as your storage of excretory waste. Everything is inside students. Everything is inside there. Okay. Excretory waste is also present. Osmoregulation is inside the vacuole. Now, students, can anyone tell me paramecium is which type of protist? If you did not tell the right answer, I'm complaining to Gopika ma'am tomorrow. Today only. I'll call Gopika ma'am and tell, ma'am, your students are not understanding. Your students are not answering in my class. Tell me, which type of protist is paramecium? All of you. Which type of protist is it? You're supposed to tell me which type of protozoan it is. Which type of protozoan is it? All of you in the chat. Ciliated, sporozoan, amoeboid, flagellated, in the chat. Paramecium. Okay. So students, in the case of your paramecium, did you see something called as gullet there? Gullet. Yes. Yes or no? Gullet there. Gullet is actually a type of your vacuole only. It has a vacuole there called as food vacuole there. You might have heard about gullet, food vacuole there. It can take things inside. Same thing here. Same thing is also present here. Food vacuole. So we have food vacuole, we have the contractile vacuole and sap vacuole. Now students, students, can anyone guess which is the next organelle which we'll be discussing right now? Can anyone guess which is the next organelle we'll be discussing? It is called as powerhouse of the cell. All of you students, all of you. This particular meme, student, this particular meme, the first meme which I showed you. Right now, students, we'll be discussing about the mitochondria. Okay. Right now, students, we'll be discussing about mitochondria. Now, students, which is called as your powerhouse of the cell right now. All of you know, right? Power cell. Why, students? It is because it produces ATP. How? With the help of ETC. What is ETC? Electron transport chain. In electron transport chain, there is something called as oxidative phosphorylation. With the help of this, it can produce ATP. Okay. Now, those of you like, sir, I do not know what is this. Do not worry. Do you want to learn plant physiology, students? Do you want to learn plant physiology? Tell me yes or no. You want to know? I will teach you. Don't worry. Okay, it's coming up. So, in plant physiology, you will learn. In plant physiology, you will learn what is oxidative phosphorylation. That time you will learn this. Not now. Not now. Now, students. Now. Now we come to the entire sausage here. Okay. Okay. This is your sausage here. Looks like a sausage. Okay. Diameter, they'll not ask you all this. Okay. Remember, site for aerobic respiration. The entire aerobic respiration, right? The entire glycolysis does not happen here. Krebs cycle happens here in the matrix. Link reaction happens here. ETC happens here. So, Krebs cycle, link reaction, ETC. All three happens here, students. So, but where do you think glycolysis happens then? If glycolysis does not happen here in your sausage here, where do you think it happens? The entire glycolysis in the chat. Okay. Now, students, it is complete double membrane structure. It divides by fission. It divides by fission here. Okay, fission. Anyone who can tell me, students, where do you think the entire glycolysis takes place? Cytoplasm. Easy. Cytoplasm, students. Okay. Now, students, this is your entire mitochondria right now. Mitochondria, can you see here? Double membrane structure here. It has the outer membrane and inner membrane here. It is also made up of lipid bilayer. It is made up of lipid bilayer, students. Lipid bilayer, right? Lipid bilayer. Okay, students. Now, this is the structure given your NCRT. This is structure given your NCRT. Outer layer, inner layer made up of crista. Inner layer made up of cristae or crista here. Inner layer. This is my diagram right now, students. This is my diagram. Okay, students, you can see here it has the outer membrane here. Outer membrane, which is much more permeable. It can allow things here. It is because it has porins in it, because it has porins, that is why it is much more. Okay, what is glycolysis? I'll teach you in the RIP chapter. Okay, respiration plants. Okay, so we have outer membrane here. This is called as your inner membrane. Inner membrane has foldings inside. It has foldings inside, students. Inner membrane. Entire inner membrane, the foldings are called as what? Cristae. Why? Because increases surface area. It can increase surface area here. Okay. Now, students, this entire mitochondria is very special. Entire mitochondria is very special. Why? Because mitochondria has its own DNA. It has its own ribosomes. So you might be wondering, which type of ribosome you can find here? See, I told you in the case of prokaryotes, we have 70S ribosome. Eukaryotes, 80S ribosome. Right? But mitochondria, students, mitochondria has 70S ribosome, just like your prokaryotes. Just like the prokaryotes, it has 70S ribosomes. 70S ribosome mitochondria. Even in chloroplast also. Okay? It in the matrix here. It has DNA, double-stranded DNA. It has, but without histone proteins, just like prokaryotes. Just like prokaryotes. Okay, it has 70S ribosome. It has double-stranded DNA without histone, just like prokaryotes. Okay. Now, apart from all the students, in the mitochondria, there are multiple racket-like structures or mushroom-like structures. Okay, right. These are students. Here we have mushroom-like structure here. The mushroom-like structure is called as what, student? This one. All of them throughout here. These mushroom-like structures are called as elementary particles or oxosomes or F0, F1 particles or ATP synthase. Every single meaning is same to same. They are referring to a single entity here called as your mushroom-like structure. Mushroom-like structure here called as ATP synthase. And students, because of this ATP synthase, it can produce ATP also. It can produce ATP because of this structure itself. And they're present, students, where? They're present in the inner membrane here. They're present in the inner membrane. That is called as cristae, students. Okay. So ATP synthase, F0, F1 particle, oxosomes, as well as ATP synthase. Everything meaning is same, elementary particles. Okay. So here, this is your entire cristae here. It can have the elementary particles of this. That F0, F1 particle. The F0 is called as the base, F1 is called as the head region here. Head, stalk, base throughout the mitochondria. This is your ATP synthase. ATP synthase right here, students. ATP synthase right here. Okay. ATP synthase right here, students. Okay. Now, students, see this. I laughed at this. Students, I don't know if you know this movie, then you can. It's called as We Are Millers. It's a movie in English, right? His main dialogue is, "Are we getting paid for this everything?" So it is based on that meme. I don't know if you're able to understand this, but yeah. Mitochondria is like, do we get paid for this? Okay. And this is your obviously salt bay putting all the ATP. Salt bay pouring all the ATP, students. Okay. Now, next organelle is called as plastid. Okay, plastid. Plastid is very special because it is found only in plant cell, and it is also double membrane. It is also double membrane, just like mitochondria. Just like mitochondria, right? It is your double membrane structure here. It is actually double membrane structure, students. Now, students, plastid can have pigments in it. Pigments in. And based on which pigment is here inside, we can get a color. We can get a color. Okay. Students, can anyone tell me one example of a plastid? Students, CS students, this is a NEET channel. NEET channel doesn't have CS, no? NEET doesn't have CS, right? Students, quickly in the chat. Can anyone tell me any name of a plastid? Any name of a plastid? All of you? Any name of a plastid? Chloroplast, 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 Chlorophyll is the pigment, Harini. Students, we have the chloroplast, where we have chlorophyll. Carotenoids are the pigments present in chloroplast. It can trap the light to do photosynthesis. Then there is chromoplast. Students, chromoplast. Chromoplast will have carotenoids that are fat soluble in nature, right? Here we have carotenes and xanthophyll here. They can impart color like your yellow, orange, red colors. Okay. Then we have something called as leucoplast. Leucoplast is also a type of plastid. See, leucoplast, chromoplast, chloroplast. Okay. While leucoplast does not give any color. No color, like no color. I will not give color only. No color only, students. But it helps in storage. For example, here we have amyloplast, ilioplast, and eluroplast. Now, students, I will teach you the way you can understand this. Okay. Now, all of you know there is something called as two types of, right, starch. All of you know there is amylose and amylopectin. How many of you know, students? How many of you know amylose and amylopectin of starch? Biomolecules class 12 chapter. Biomolecules class 12 chapter you learn in chemistry. Amylose and amylopectin. See, amyloplast, amylopectin. So storage of starch or carbohydrates. So it helps in storage of starch or carbohydrates. Okay. Now, oils and fats. Eluroplast. See, all of you might know that in case of monocot seed, there is something called as aileron layer. Right? Aileron layer, students. Aileron layer here. Aileron layer. They help in aileron layer, protein layer. So it helps in storage of proteins. Okay. So their main function is storage. They are no color here at all, colorless. Okay. Right. Their main function is storage. They are present inside the cell. These are present inside lover in ruins. Students, tell me your real names. Real names. I can call you real names. Okay. Genetics I'll teach you, don't worry. It's coming up. So we have amyloplast, ilioplast, eluroplast, storage of things, students, storage of things. Okay. This is the structure. This is the structure. Can you see? Structure of a chloroplast here. Outer membrane, inner membrane here. Outer membrane, inner membrane, students. Okay. Now, students, I'll just show you one 3D image right now, students. 3D image. Chloroplast here. And I'll teach you some little bit extra also. Students, see this? This right here is a chloroplast here. This right here is a chloroplast right here. Okay, this right here is a chloroplast. Ishwar, okay. Ishwar, see this. In this chloroplast, can you see double membrane, outer membrane, inner membrane here? Okay. Now, students, in this chloroplast, all of you can see something called as coin-like structures here. Can you see, students? There are multiple coin-like structures here. Coin-like structures. Now, these coin-like structures are called as thylakoids. They are called as thylakoid right now. Okay. They are called as thylakoid right now. Thylakoid. These coin-like structures are called as thylakoid. Okay. When students, when this entire thylakoid, they form a stack. When the coins become a stack here, this entire stack is called as grana. Called as grana. Then students, one stack, second stack, third stack, fourth stack. Throughout the chloroplast, we have multiple stacks here. Okay. Now, students, in this stacks itself, right? In this thylakoid stack called as grana, we have the pigment called as chlorophyll. So chlorophyll is present here itself. Okay. Now, students, can you see between this stack and this stack, there's a connecting link here. This is called as stromal lamellae. Okay, it's called as stromal lamellae. Okay. Now here we can find its own DNA, we can find own lipids, we can find own ribosomes here. 70S ribosome, just like your prokaryotes. 70S ribosome, DNA without any histone here. Coins called as thylakoid. Entire stack is called as grana. Connecting link is called as stromal lamellae. Now, students, it has a matrix here, students. Entire matrix here. Can all of you see, students? Matrix here. Matrix. The matrix is called as stroma. Matrix is called as stroma here, students. Okay. The matrix is called as stroma here. One second, students, 1 second. Matrix is called as stroma here. So here I want all of you to understand this grana, entire stack, thylakoid, stroma lamellae, the connecting link. In all of these three things, we have the light reaction taking place. Light reaction taking place here. Light reaction. Okay. While the stroma here, the matrix here, entire dark reaction happens here. Now, what is light reaction, students? Light reaction, basically trapping of light happens. In the dark reaction, the glucose is produced in dark reaction. This I will teach you in plant physiology, photosynthesis in higher plants chapter, which is my favorite chapter of all time. Okay, yes, students. See here, chloroplast, I use light energy to make glucose. Mitochondria, I break to make ATP. This is how it functions inside a cell. Inside a cell, this is what happens. Light energy is trapped by your chloroplast glucose. Entire glucose is used by mitochondria to make ATP. This is what we learn in your first plant physiology. This is what we learn in photosynthesis. This is what we learn in respiration. In respiration. This is the two chapters of plant physiology. This is the two chapters of plant physiology right here. Okay. Now, students. Next thing is called as ribosomes. Okay, ribosomes. Okay. There are small structures here throughout. You see here in the inside here, small structure called as ribosomes here. It was discovered by George Paldane. What are they made up of, students? They are made up of RNA and protein. So ribosomes are made of what? Ribosomes are made up of RNA plus protein. This is what they are made up of. This is what they are made up of, students. They do not have any membrane here. In eukaryotes, they are 80s. Prokaryotes, they are 70s. 80 is made up of 60 plus 40, while prokaryote is made up of 50 plus 30. This is called as your large subunit. This is called as small subunit. Small subunit, students. Here the S is called as Sedgwick unit, also called as sedimentation coefficient. Also called as what? Sedimentation coefficient, students. Also called as sedimentation coefficient. So you can see here also. Small subunit, large subunit, complete structure, prokaryotes. Small subunit, large subunit in the case of your ATS, in the case of your eukaryotes. 60 plus 40 is 100, but ATS here because of the sedimentation coefficient. Okay. This is your 2025 NEET PYQ also. 2025 NEET PYQ right here on the board. Okay, students. Okay. Now, students, next concept we are coming towards the students. Right now, we are coming towards the end of the chapter, right now, students. Almost the end of the chapter is coming up. Okay. Next thing is called as cytoskeleton. Again, till here, the entire chapter is very, very easy. 99% of students do not understand what is cytoskeleton. They do not understand what is cilia. They do not understand what is flagella structure. Students, if you are ready to learn that and not be that 99% students who don't understand anything, okay, only few students understand the structure of cilia and flagella, okay, and today I will be teaching you and I'll be making sure you are part of the top 1% who understand the structure of cilia and flagella. Students, all of you ready? Students, ready in the chat? Can I get ready in the chat? Students, all of you ready? Ready to understand what is cytoskeleton, what is actually the structure of cilia and flagella, and how they make up the entire structure. Students, all of you ready? Yes? All of you. Ready, students? Ready, all of you? Yes? Sunday test revision, huh? Okay. Students, we have a lot of DNA batch students right now. We have a lot of DNA batch students in the chat also, because they have a test coming up this Sunday. This Sunday they have a test on this topic, so they are in the comment section to learn, revise one more time. I feel all the DNA students. Okay, now, students. First of all, what do you need to know about cytoskeleton? All of you need to understand here that just framework, bones. In the case of a cell also, there is a framework here. The framework made up of proteins That is called as cytoskeleton, students. That is called as cytoskeleton. That is called as cytoskeleton, students. This is a framework. Framework here. Okay. And the cytoskeleton is made up of proteins. It is made up of protein, students. Protein. Okay. Basically, it is a network inside. It is a network or filaments scattered inside a cell. Okay. Let me just show you. Let me just show you here. This is how they are. Can you see, students? Different types of cytoskeletons here. Okay. Inside a cell, they are scattered everywhere. There is different types of cytoskeleton present inside a cell here. Different types of inside a cell. Okay. Now, students, there are three types here. One is called as microtubule. One is called as your microfilaments. Third is called as intermediate filaments. Three types of cytoskeletons here. Okay. 1, 2, 3. Now, students, here majority of your questions will come from microtubules itself. Majority of your organs like centrioles, spindle fibers, and as well as the cilia and flagella, they are made up of microtubules only. Okay, they are hollow, solid, hollow, unbranched, non-contractile. It means they are very rigid. They cannot bend. They cannot bend here. Okay, they are made up. They are present. These microtubules, they are present in cilia, flagella, and centriole. So cilia, flagella, and centriole, all three are made up of what? All three are made up of microtubules itself. Okay. Then we have your intermediate filaments and microfilaments. These two not so important. We'll be focusing on microtubule more right now. Microtubule more, students. Now, what are the functions here, students? What are the functions here? There are three main functions: mechanical support, motility here, it's a PYQ, and maintenance of shape of the cell. Three functions of your entire cytoskeleton here. Cytoskeleton. Okay. Remember again, microtubules, microfilaments, intermediate filaments here. Here, microtubules are hollow, unbranched, non-connectal. This they might not ask you, students. This is very important. Cilia, flagella, and centrioles are all three structures are made up of microtubules. Okay, students. Now we come to cilia and flagella. I want all of you to pay extreme attention right now. Extreme attention. Okay, extreme attention right now. Okay. What is cilia and flagella? Okay. Now, students, did we hear about flagella in your prokaryotes? Tell me yes or no, student. Tell me yes or no. Did we learn about flagella in your prokaryotes? Yes, we did. Yes, we did. There we had something called as your basal body, hook, and filament. The flagella of prokaryote and the flagella of eukaryote, they are two separate entities. They do not look like at all. They're very, very different from each other. Very different from each other, actually. Very different from each other. Okay. Here, basically, the students, their hair-like outgrowths on the cell membrane, they emerge from the basal body here. Let me teach you, students. Let me teach you. Okay. So, students, this is the diagram given your NCERT. This is the diagram given your NCERT, the diagram of flagella and cilia. Can you understand anything from it? Students, can you understand anything from it? Tell me in the chat quickly. Students, do you understand anything from here? Tell me yes or no. Tell me yes or no, students. Tell me yes or no. Just by looking at this diagram, do you understand anything about cilia and flagella, how it looks like? All of you in the chat. And if you're new to the channel, students, quickly smash the like button, all of you. Can you understand anything from here? No, not at all. Let me teach you. Let me teach you right now. Let me teach you. How does the cilia and flagella actually looks like? Students, this right here is a flagella. Students, this right here, students, this right here is a flagella. This right here is a flagella, this flagella. Okay, flagella. This will move around. It will move around. It can move. This is a flagella right here. This has a bottom part here. Can you see this bottom part here? This bottom part is called as basal body. Basal body here. From this, we can see a flagella rising here. Now, students, if you see a cross section of top view, you take a cross section and you look from the top side. This is what it looks from the inside here. Okay. Now students, all of you tell me in the chat, don't you think so? These structures look like hollow tubes. Students, tell me, this top view section of a cross section of a flagella, doesn't it look like hollow tubes? Tell me yes or no, student. Tell me yes or no. Yes, they are basically hollow tube like structures. They're basically hollow tube like structures, and they are made up of what? They are made up of microtubules. They're made up of what? Microtubules. So this is a flagella here, long flagella which has plasma membrane outside, and if you see a top section, cross section, this is how it looks from the top view. Okay, now if you zoom in, this is how it looks. Okay, this is how it looks from the outside. Top view, same top view. Okay, same top view. See, cross section, top view. This is what it looks from the top view. Now students, let me just show you one more diagram of mine. Okay, before we go, students, let's just understand what is cilia and flagella first of all. Understand what is cilia and flagella, students. Understand what is cilia and flagella right now. Okay, cilia is much more shorter and smaller. Flagella is longer here. It has oar-like movement. What do I mean by oar-like movement, students? If you are ever anytime gone in a boat, these structures are called as oars. They are called as oars here. Now let's say you hold the oar and you move it. Will you go front? Yes. Move it back, you'll go back side. So depending upon the oars, you can move front or back. Similarly, how this oars play a major role in pushing the water. Same way, cilia, which is present throughout the body here. You know, see small here, small cilia. They're present throughout the body. They will push the water. They'll push the water and this entire organism can move around. That is what is seen in the case of your paramecium also. Paramecium has a cilia throughout it. That is why paramecium is part of ciliated protozoan. Cilia. So this entire cilia will push the water and this will move. Same thing happens in the case of the movement of egg also through the fallopian tube. Ciliated movement here. Okay, students, see here. Entire thing throughout the body, tiny cilia throughout the body. Then at the end we have flagella. Long flagella at the very end, students. Long flagella at the very end. Now students, this is the diagram of mine. This is a diagram of cilia or flagellum, right, students? Cilia, flagella. And this is a diagram from the cross section from top side. Okay. Now students, if you notice here, it has something called as, outside it has the plasma membrane on the outside. Okay. Now in the center, what do we have? In the center also we have two separate central microtubules we have. In the center we have two central microtubules we have. Yes, we do. On the periphery, students, on the periphery, what do we have? On the periphery we have peripheral microtubules on the outside. Okay. On the outside we have the peripheral microtubules. And all of these peripheral microtubules are present as doublets. They're present as doublets here. Can you see, students? Doublets. So they have peripheral microtubules on the outside. Okay, peripheral on the outside. And students, these are hollow cylinders. Hollow because they're microtubules. They're hollow in nature. Hollow. So central 2, outside 9. 1, 2, 3, 4, 5, 6, 7, 8, 9 here. Okay. Now students, this entire peripheral microtubules are connected to each other by something called as linker. This orange part is called as linker. Linker. Okay. Now students, here this pink color part is called as central sheath. Now every single peripheral microtubule, right, from this doublet, the A part will have a spoke coming out like this. This spoke is attaching to the central sheath. This spoke here is called as radial spoke. Okay, the spoke is called as radial spoke. This radial spoke is connected to the central sheath, and that is the entire thing is called as your cilia flagella. The cilia flagella in the right in the middle, it has a bridge also, and this entire middle part is called as core or axoneme. This entire part is called as what? Core or axoneme. Okay. Now here we saw 9 peripheral microtubules. So 9, central 2 plus 2. So here we can, students, here we can find 9 plus 2 arrangement here, students. Here we can find 9 plus 2 arrangement of microtubules. 9 plus 2 arrangement of microtubules. 9 plus 2 arrangement of microtubules can be found here. 9 periphery, 2 central. Right? 2 central, students. Okay. 2 central. Students, 1 second, students. Okay. So this is called 9 plus 2 arrangement. Students, tell me, now does it make sense to all of you? Students, now does this diagram make sense to all of you? In the chat quickly. See here. Here we have the plasma membrane on the outside. Here we have the linker, basically the interdistal bridge, the linker here between those two. Here we have the central microtubule here. Here we have the central microtubules here, 2 central, that is the 2. Okay. Peripheral microtubules here, doublets, 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 plus 2 arrangement. 9 plus 2 arrangement of microtubule, students. Akshaya, did you understand, Akshaya? This is a top view cross section of a entire cilia and flagella. Both will help in swimming. Can you see bridge here? Green color part is called the bridge here. Okay. Middle part is called as the bridge here. 2 central microtubules, 9 peripheral microtubules here, connected to each other by a linker here. Okay. Then we have the radial spokes. Can you see, students? Radial spokes coming out here. Radial spokes are connected to what? Radial spokes are connected to the entire central sheath. So this, students, this right now. See, this is different, students. This is called as inter-doublet bridge. Inter-doublet bridge is similar to linker. It is similar to linker. Okay. This is the middle bridge here. Okay, middle bridge. One more bridge here. This is similar to linker. Not exactly linker, actually, they are different from linkers, but it is similar to linker itself. Inter-doublet bridge. Students, how many of you understood the structure of cilia and flagella? If you just learn this from your NCERT, very difficult to understand. You need to visualize this. This is your basal body. Basal body is called as right? Kinetosome. From this we have the flagella coming out here. Flagella cross section, top view. If you see top view, we can see 9 peripheral microtubules, 2 center radial spokes, central sheath here, and linkers everywhere. Linkers, students. This diagrams you forget. This all is extra information. Students, this is called as the structure of cilia and flagella. If you learn from the NCERT, God promise, very difficult to understand. Okay, students, if you understood it, quickly fire emoji in the chat, all of you. See here. PYQ. Which of the following is a function of cytoskeleton motility? Yes, students. Now, students, now. Again, if you didn't understand, I want you to watch the lecture one more time. Please watch the lecture very slow one more time. Draw the structure. It's very beautiful to understand biology. Okay. Now, students, understand centrosome and centriole also. This entire structure, students, centrosome is one entire structure here. This entire structure is called as centrosome. Entire structure is called as what? Centrosome. Now, inside one centrosome, we have 2 centrioles. See, centrosome contains 2 cylindrical structures called as centrioles. So, one centrosome has how many centrioles? 2 centrioles, 1 centrosome. Okay. Now, one centrosome has 2 centrioles which are perpendicular to each other in a 90 degree to each other. Okay. 90 degree to each other. So, one centrosome has 2 centrioles. 2 centrioles here. Now, this does not have membrane. Students, I've told you before, it does not have any membrane here. Okay. So, it does not have any membrane. Actually, it has membrane-like structure here. The membrane-like structure here is called as what? Amorphous pericentriolar material or amorphous pericentriolar space. Amorphous pericentriolar space here, outside. Okay. Now it is also called as kinoplasm or centrosphere. The other names are here. Now students, I will teach you right now something called as structure of a centriole now. This one centriole. Understand? Students, see this. Two centrioles perpendicular to each other. Students, can all of you see there are hollow structures here? Hollow structures. Hollow. So if there are hollow structures in a cartwheel shape, can I call them, students, this hollow structures, can I call them microtubules? Yes, they are microtubules again. They are microtubules. Okay. So students, here is my diagram first of all. This is my diagram here. Okay. Here also, students, here also, outer part is amorphous center space here. Here also, students, we have peripheral microtubules. Where students? Here we have the microtubules, peripheral microtubules. Okay, peripheral microtubules on the outside here. Now these peripheral microtubules here are present as triplets here, but triplets 3, 3, 3, 3, 3. Now students, I want all of you to count and tell me. All of you want to count and tell me, students, how many do you find in periphery? All of you. 1, 2, 3, 4, 5, 6, 7, 8, 9. We find 9 periphery microtubules here. 9 peripheral 9. Now students, here all the peripheral has something called a spoke coming out, called as radial spoke here. Between two peripheral microtubules, we have linker in the middle. Okay, linker in the middle. Okay. Now I want all of you to understand. I want all of you to understand here, right? This peripheral microtubules, right in the center, it does not have anything. It does not have any microtubule. Gopika ma'am, 10 minutes. 10 minutes, Gopika ma'am, 10 minutes. Give them a break. Ma'am, I'll join. 10 minutes. Okay. Now students, here in the center, it does not have microtubule. In the center, it has a protein structure called as a hub. This one. In the center, it has a protein structure called as hub here. Protein structure called as a hub. Okay. So if inside there are no microtubules, can I tell 9 plus 0 arrangement here? All of you see 9 peripheral microtubules which are triplet. Between them we have linker here, radial spoke connecting here. Right in the middle we have something called as your hub, which is 0 because there is no microtubule in the center. We have a hub here. Okay, we have a hub here, students. Okay, so we can see 9 plus 0 arrangement here, students. 9 plus 0 arrangement here. Okay, so this is the structure of your entire centrioles. See here also, students, see 9 peripheral microtubules, triplets. Okay, see here also, students. Top view, centrioles. 1, 2, 3, 4, 5, 6, 7, 8, 9. Triplets. Okay, triplets everywhere, students. Okay. Now, students, this entire structure is similar to basal body in the cilia and flagella. See here, students. If you go back here, I told you basal body. This basal body also has a structure similar to 9 plus 0 here, and this top part is 9 plus 2 arrangement here. Cilia and flagella. Basal body is similar to your centriole structure. It is similar to centriole structure, that is 9 plus 0 arrangement. 9 plus 0 arrangement, basal body. Top part is 9 plus 2. 9 plus 2 arrangement. Okay, students. Friends, can you see here? Basal body, 9 plus 0 arrangement. Top part, 9 plus 2 arrangement. Cilia and flagella. Okay, the centrioles form the basal body of cilia and flagella. Okay, basal body of cilia and flagella, students. Okay, no, students, no. Students, now we come to the last part here, right? Last part of this chapter. After this chapter is over, also, right? Nothing is left. Okay, that is called as nucleus. Okay, that is called as nucleus, students. Right? Now, nucleus, students, can anyone tell me who discovered nucleus? Anyone? Can anyone tell me who discovered nucleus, students? Anyone? Who discovered nucleus? Anyone? It was Robert Brown. Correct. Once again, students. Okay. Okay. So students, it was discovered by Robert Brown. It was discovered by Robert Brown here. Fleming, nucleus stained by basic dye. So he discovered, students, Fleming actually discovered the chromatin. Chromatin. Okay, chromatin. Now, what is chromatin? What is nucleus? I'll teach you all of it. Listen to me, students. Okay. Now, I want all of you to understand this diagram. If you understand this diagram, everything is clear, students. Okay. If you understand this diagram right now, right, everything will be clear to all of you. Okay, see this, students. See this. Okay. Now, I want all of you to understand, students, here. A nucleus has how many membrane? A nucleus has two membrane. So all of you see, students, it has outer membrane here and it has the inner membrane here, students. Outer membrane, inner membrane. Between the two membranes, the space is called as perinuclear space. Between the two membranes, it's called as perinuclear space, students. Okay. Now, can all of you see, students, from the outer layer, we can see the ER here. ER coming from the outer layer here. ER. Okay. Now, students, here in this between the outer membrane and inner membrane, in the perinuclear space, there are small gaps here. Gaps. The gaps are called as what? Nuclear pore here. Nuclear pore. Function: the nuclear pore are a passage for the entry of your mRNA. Why? Because see, translation happens inside the nucleus. So mRNA has to go out. It'll go out through nuclear pore present in this here. Nuclear pores everywhere. Okay, nuclear pore. Now, students, on the inside the matrix is present. The matrix on the inside is called as what? It is called as nucleoplasm. Nucleoplasm is the matrix, students. Nucleoplasm is the matrix on the inside. Okay. Now, students, inside the nucleus, there is genetic material. All of you should remember, in the case of cell, we generally do not find chromosomes. Chromosomes are found only during cell division. But in normal case, we always find the chromatin. We always find chromatin, students. So chromatin is found where? Chromatin is found in your resting phase always. Resting phase, we find chromatin. What is chromatin, students? Chromatin is basically DNA plus histone and non-histone proteins plus RNA. That is your chromatin. So chromatin is like a long thread made up of DNA. Okay, that's what inside. So we have chromatin, thread-like structure inside. We have nucleoplasm is the matrix here. Now, students, here we have something called as round structures here, free floating. They are called as, students, nucleolus. Nucleolus. Now, what is the function, students? The main function of nucleolus is that they produce rRNA. They produce rRNA, students. So, I'll just repeat one more time, students. Outer membrane, inner membrane, perinuclear space in the middle. In this, we have nuclear pore, chromatin inside. We have the nucleoplasm on the inside. Chromatin is basically genetic material here. Then we have nucleolus here, which produces rRNA here throughout. That is the entire structure of nucleus. So, students, you get the notes, you'll understand very easily. Okay. Students, can you see here? Nucleolus, chromatin coming outside, students. Chromatin coming outside. Okay. Students, there are certain cells like your RBC and as well as your CU plates. they do not have any type of nucleus. They're like, I have given up my nucleus. They're like Dana, they're like Karna, they'll give away their nucleus. They do not have nucleus only. Both of them do not have any nucleus at all, students. Nucleus at all. Okay. So this is chromatin, students. This is chromatin here. Chromosome has coiling of chromatin here. Chromatin undergoes condensation to give chromosomes. Okay. Now, students, this is a typical chromosome. A chromosome has something called as a primary constriction and secondary constriction. See, this right here is a primary constriction here, students. This right here is a primary constriction here. Primary constriction. Primary constriction is called as centromere right in the middle, centrome here. Centromere is generally inactive. These two structures are called as sister chromatids. They are called as what? Sister chromatids here. So one chromosome can have one chromatid or two chromatid. Okay. Now here we can see spindle fibers attaching. Friends, remember, spindle fibers will always attach to kinetochore, not to centrome here. See, this is centrome here. Around centrome here there is something called as kinetochore. So all the spindle fibers will attach to the kinetochore itself. Okay. Now, friends, there is something called as secondary constriction here on top here. Okay. Secondary constriction is called as satellite. It is active in nature. It is active in nature, students. Okay. It is made up of, out of all the 23 chromosomes, 5 will have satellite. Okay. Now this ends of chromosome is called as telomere. Ends of chromosomes are called as what? Telomeres here. Telomeres. So primary constriction, secondary constriction, telomeres at the edge here, kinetochore where the spindle fibers attach, not the centrome here, and sister chromatids. Okay. Now here, students, you can see it has P arm and Q arm. Top part is called as P arm always, bottom part is called as Q arm. Q arm. This is your Q arm, this is your P arm on the top. Okay. So depending upon the arm length, P arm and Q arm, right? P arm and Q arm, there can be different shapes of chromosomes. Okay. There is metacentric, submetacentric, acrocentric, telocentric. See this. See this. Can you see, students? In this, one arm is missing completely. One arm is very small. One arm is in some almost equal. Here it is equal. Okay. So based on the different chromatid length or arm length, they have different types of your shapes of chromosomes. Shapes of chromosomes. Shapes of chromosomes. This is your NCI diagram. NCI diagram given right here, students. NCI diagram. Okay. Now last topic is called as microbodies. Microbodies are small structures, vesicles present in a plant cell and animal cell. Here we have something called as peroxisomes, right? Glycooxysomes. Two types are there. Nothing much. 2 lines you can read, all of you students. 2 lines you can read. That's all the chapter, students. That's all the chapter. Okay. Students, there are multiple questions here. All of you can solve the questions. Since the time is very less, right? Notes, I'll give you both the class notes together. I'll compile and give it to you, right? So students, I will see you in the next class. Until then, take care all of you, right? If you're not a part of DNA batch, join DNA batch right now, because DNA batch, this all is completed. Even more detail I've got in DNA batch. Okay, students. So thank you so much, students. Bye bye. Take care all of you, and I will see you in the next class. Until then, take care and bye bye all of you. Bye bye.